So we are right on time. Welcome to our participants who are slowly streaming in to this live webinar. I think we are ready to start. We can go ahead. So hello everyone, good morning, good afternoon, good evening, depending on the part of the world that you're joining us from. We welcome you to this webinar as the allies of science. It's a live webinar that has been organized by the Alliance for Science, and we're glad to have such a huge number of participants joining us. Thank you for creating the time to be with us. Today's webinar is a part of a series of monthly webinars that are hosted by the Alliance for Science, and it seeks to promote access to scientific information as a means of enhancing food security, improving environmental sustainability, and raising the quality of life globally. We are excited for today's webinar series, where we unpack a very key and pertinent question: is Kenya ready for gene editing technology and products? This is indeed a marvelous time for us as scientists, especially those that come from the field of genetics, due largely to the advances that are there when it comes to genetic analysis and gene manipulation. And this question is particularly more important because we're seeing there's an advancement, especially in gene editing research, with the use of CRISPR-Cas in research globally, as well as here in Kenya and across Africa. We are seeing that these tools are accessible to all of us, and they offer a wide range of benefits, which we'll be hearing from our panelists. For this webinar today, we have two researchers that have been working on gene-edited technologies around the area of agricultural biotechnology. We also have a key national regulator who will be sharing the insights when it comes to the regulatory landscape. For those who may not know, my name is Doris Wangari. I'm currently a research scientist at KEMRI, which is a Kenya Medical Research Institute here in Kenya, and I hold about 10 years experience when it comes to biotechnology. I'm very passionate around the regulatory processes, and I hold a very keen interest as well on gene editing, and I'm happy to moderate this session for you today. Before we start, we have two key housekeeping rules that I'll be sharing with you. One is that this webinar is being recorded and will be made available on all the social media platforms. That is where we have the @ScienceAli on Twitter, Instagram, as well as Facebook. We are currently live on Facebook as well. So thank you for the participants that are joining us from that platform. Additionally, the webinar is 1 hour long, and we envision for it to be an interactive one-on-one session. So sadly, there will be no presentations. We therefore ask for our participants to send in the questions to the various panelists that we have, or add in some of the comments or insights around the various channels of communication that we have. So without further ado, let us get into it. I want to introduce the range of panelists that we have here today. And as the old adage goes, ladies first. Ladies and gentlemen, our first panelist is Dr. Leena Tripathi. She's a principal scientist in plant biotechnology at Iita, which is the International Institute of Tropical Agriculture, where she serves as the Deputy Director of East Africa Hub, and she's also the country representative. She has been involved in plant biotechnology research for more than 20 years, with specific interest when it comes to crop improvement. Thank you for being here, Leena. Our second speaker will be Professor Stephen Runo. He's a Kenyan researcher at the Kenyatta University, where he serves as an associate professor of molecular biology. Runo has extensively studied the pathogens of African cereal crops, including striga, and he's also a Royal Society African Prize winner. Welcome, Steve.
Thank you, Doris.
Thank you. Finally, our third speaker will be Mr. Josphat Muchiri, who's the Deputy Director, Technical Services at the National Biosafety Authority. He's a regulator who is in charge of the research and commercial activities, and he has a view to ensuring safety to human and animal health and provision of adequate level of protection of the environment. Welcome, Josphat. You're muted.
Okay, sorry. Thank you very much, Doris.
Karibu. Karibu means welcome for those participants that may not know Swahili. So thank you all for making the time to be here. So we'll start off with a bit of getting to know each one of you. And I'll start off with Leena. As I introduced you, I said you have about 20 years of background in plant biotechnology. So what motivates you to go to the lab each day, and what made you decide to focus in biotechnology?
Thanks, Doris, for your kind introduction and the question. So let me answer your second question first. What motivates? And actually, I started biotechnology long back during my master's. So I did the master's in biotechnology and genetic engineering. And I did that from India. Actually, I was the third batch when Indian government started giving masters in biotechnology. And my always the motivation for doing biotechnology and genetic engineering was actually how the agriculture production can be enhanced for in order to reach the food security. So that was the whole idea. And then initially I did the biotechnology like more as a broader aspect, like not only agriculture, but agriculture plus the human health. So I did quite a lot of work on immunology during my master's. But then I have confined more to the agriculture. So I did plant biotechnology. And I have been working into the plant biotechnology for more than 25 years. I think the introduction you have is like few years old. Currently, I'm the director for the East Africa and based in Dar es Salaam. So what and then, like, you know, when for me the best part of my day is like, you know, when I go into the lab. And it's still, even though I have a lot of administrative responsibility, but I still try to go to my lab almost every day after lunch. And all my group knows that, you know, I'll be there after lunch. And then the best part is, like, you know, when I have interaction with the students, with the researchers there, to see what challenges they are facing, how we can resolve those challenges. And the next day when I go, and if I see some result which they were facing the challenges of previous day, that's it, you know, that like boosts my energy. And then, you know, that's the something which I will say is the best part of the day for me. Yeah.
Wow, thank you for that. Indeed, when you become a science and become, I said, when you're a scientist and also an administrator, it poses a challenge when you get the time to enter into the lab. But yeah, the purpose that you have and the drive to ensure that you enter the lab daily in the afternoons, yeah, indeed, it's enviable. For the next question, I'll pose it to Professor Runo. I'll also ask you a similar question, but more around how did you enter into this field of biotechnology, and how does it relate to genome editing research?
Yeah, thank you, Doris, and thank you, participants. No, I did not enter into biotechnology by accident. Deep down, I'm a botanist, I'm a plant biologist. I've always been fascinated by plants. And so beyond my fascination, then I went to school to study biochemistry at undergraduate, and then molecular genetics of plants at master's, and then plant molecular biology at PhD. And I worked with some of the finest plant biologists, including Professor Jesse Machuka, who would be very familiar to a lot of people in this audience, Professor Andrima Sinha, Davis, and Professor Peggy Lamore. Later on, after PhD, with Professor Julie Scholes and Professor Mike Timko. So really got to be exposed to the depth of plant biology, which is my passion. And really the goal was to these plants that I'm really fascinated with, how can we make them better and better in the context of how can we improve them to increase yields, and very specifically to help increase food and nutritional security in the continent that I live in and the continent that I love so much, Africa. Thank you.
Wow, thank you for that. Thank you for that, Prof. But for the background that you've shared, I'll ask you the next question, Leena. You are currently doing some research around gene editing. So what are you working on right now? And as well, why did you turn to genome editing instead of the other technologies that we have?
Thanks, Doris, for your question. So currently, I'm applying the gene editing for the improvement of banana, which is a very important crop for Africa. You know, banana is grown more as like a fruit crop worldwide, but then when you come to Africa, actually is a very important staple food crop, and particularly for the East Africa, because in many countries like Uganda, Rwanda, you know, the consumption of banana is much more than any cereal crop, you know, so that's the major staple food for them. But being said that, you know, it has a lot of production constraint, particularly diseases and pests. And most of the time you will find that, you know, bacterial disease, fungal disease, and even the viral disease, and maybe some of the pests like nematodes, they are coexisting. So that means they are present in the same field at the same point in the farmer's field. So farmers are really having a lot of challenges with the banana production. So what we are looking at is, like, you know, if we can develop a broad spectrum resistance for more than one pathogen and pest. And if you apply a traditional technologies, that takes long time, because, you know, you have to one by one stack those traits. Sometimes you will find that, you know, resistance to those pathogens are not even available in the germplasm. So that means, you know, you cannot apply, you know, if one of the parent has resistance for the resistance, then you cannot cross the two parents. I give you example, like for the Xanthomonas wilt, the bacterial disease, which is one of my focus. All the cultivated bananas are susceptible. So that means there is no resistance in the germplasm. But with the long studies in my lab, we find out that, you know, there is resistance into the wild progenitor of this banana, which is called Musa balbisiana. So originally bananas came from Musa balbisiana and Musa acuminata, those two wild types. So, you know, we studied in detail the molecular basis of the resistance to the Xanthomonas wilt in this Musa balbisiana. And now genome editing is a very powerful tool which we can apply just to make some like a very targeted, precise, small changes into the banana genome instead of bringing any gene from outside. And so it's just like simply tweaking little bit the banana genome to make it resistant. So now we are actually transferring the knowledge from the wild type banana into the cultivated varieties, which are preferred by the farmer. So we are not actually, so the varieties which farmers love to grow, but then they are not managed to grow right now because of the disease. So we are trying to actually develop the resistance into those varieties based on the information what we have in the wild type. And normally in the banana breeding, you don't cross Musa balbisiana with anything. So, you know, you cannot use the traditional technology. So that's a comparative advantage, I will say. And another comparative advantage is in the time, because if you do the crossing, you know, it's not, you cannot just cross for one gene, you know, so many other undesired traits will also come in, and which you have to back cross or cross again several times to, you know, to fix the trait and to get the desired quality of the banana. But with the genome editing, at one step, you just precisely change the genome, and then your final product is there. So that's why we are applying this technology. We started actually working on gene technology in gene editing in 2015. So it's few years we have been working on this, and we have been working on one bacterial disease, one fungal disease, which is Fusarium wilt, and also the virus, banana streak virus. So we are working on 3 traits in bananas using gene editing.
Okay, and this work is being done here in Kenya?
Yes, everything is done in Kenya at Iita, based at Beka Ilri Hub.
Okay, so thank you for that. I think what I've heard from you is that the technology is precise when it comes to the application of transferring of the genes, and it's also fast, offering a wide range of benefits. It's also specific because you're able to carry just a few of the genes that you need and transfer them. So thank you for that. The next question, I'll come back to you again, Prof, because I feel like we need to set the ground in terms of the research that is ongoing. So I'd ask you a similar question. What are you working on currently in the lab, and are you applying genome editing technology?
Thank you, Doris. So starting from the fact that our farms are really small, and the fact that agriculture in most parts of sub-Saharan Africa is carried out by smallholder farmers with very small pieces of land. And the fact that these farms are heavily infested by pests and diseases, and the realization that any additional pressure to these lands will expose millions to hunger and starvation. What we have done, or what we do, is to leverage techniques of advanced plant molecular biology and advances in plant sciences in general to see if we can be able to create better crops. And the focus of my group right now is on a parasitic weed called striga. And what striga does is that it attaches to roots of crops, cereal crops, especially maize, millet, sorghum. In fact, most of all cultivated cereals in sub-Saharan Africa are attacked or parasitized by one or more species of striga. And what our work has done, or what research we have come into ourselves and other groups, is realization that you can use genome editing to impart resistance and to significantly reduce the infestation or increase resistance or impart resistance in some of the crops. The crop that we're working on for the moment is sorghum. And what genome editing does is to provide, as you say correctly, a very specific alteration of DNA sequence. That in technical terms is what scientists call mutations. And mutation is an ongoing process. In fact, the diversity of crops and the diversity of plants and animals, and indeed every living organism, has a lot of it coming from mutations. And so what genome editing is doing, or what genome editing does, is to fast forward the process of mutation, a process of mutation for beneficial reasons, and in this case, to improve agronomic traits of crops, and for our case, disease resistance.
Thank you for that, Prof. Indeed, again, I believe the work that you're doing is for the benefit of the smallholder farmers, you've indicated, and also highlighting the plight of the striga and the need for adopting genome editing to address that in your research. The next question I want to pose to Mr. Muchiri, who is the chief regulator for this new technology in the country. And the question that I want to ask you, Muchiri, how is the regulatory landscape in Kenya currently, and how does it affect this ongoing researches that Dr. Tripathi has been doing and Professor Runo has also been engaging in?
Thank you very much, Doris. Thank you for the question. The technologies that Dr. Leena and Professor Runo talked about, they are being carried out here in Kenya. In fact, the National Biosafety Authority was established more than 10 years ago. It was established in 2010. basically to regulate issues of modern biotechnology, specifically on genetically modified organisms. But even before the establishment of NBA, Kenya started the issues of ratification, the signing and ratification of the Cartagena Protocol on Biosafety, which is basically an international treaty for regulation of modern biotechnologies. When that protocol was signed, the government started the process of domesticating at the national level. The cabinet approved the National Biotechnology Development Policy in 2006. That was followed shortly with the enactment of the Biosafety Act of 2009. So when the Biosafety Act was enacted in 2009, we realized that there were no supporting regulations to operationalize the Act. So in 2011 and 2012, the government published 4 biosafety regulations with specific frameworks for certain activities, including research. But if you look at critically at the Biosafety Act and the biosafety regulations, there are no very clear provisions on how genome-edited organisms and their products will be regulated here in Kenya. So what the board did, because in 2014, 2015, we started receiving applications from our research institutions. First of all, inquiring on whether they are covered under the regulation of NBA. At that time, the board guided that they should be regulated. All genome-edited organisms should be regulated using the frameworks of genetically modified organisms. That includes the Biosafety Act and the various biosafety regulations. That was to happen until a clear framework or clear guidelines were developed specifically for genome editing guidelines. So in the last 2 years, the authority had, in consultation with various stakeholders, including the scientific community, the government agencies, we have been able to come up with guidelines on how gene-edited organisms will be regulated in Kenya. Those guidelines were published this year in March. They were published on 15th March 2022. So currently we have the legal provisions, the Act regulations, and now more specifically, we have a guideline on regulation of gene-edited organisms. So those projects that the scientists have talked about, they are currently regulated, those frameworks that I've just talked about. Thank you very much.
Okay, thank you for that, Muchiri, for the background history of how we came up, the establishment of the NBA and the various legal instruments around that. It follows the similar story around the history of as well of genome editing, and the interest is now growing even more as well. So are there any specific differences when it comes to the regulation of gene-edited technologies versus GMOs?
Yes, and that was the reason. That was the reason for having a specific guideline on the genome editing. We realize in the gene editing process, some of the processes could lead to products that fall under what we call the definition of a GMO. And also using the same gene editing processes, the end product could be a product that resembles a conventional product, a product that do not have any foreign or novel genetic material. So because of these differences, that was why we identified there was a need to have a specific guideline on genome editing. So if you're able to look at the guidelines which we have published in the website, we have categorized these products into two. So these products, there will be products that will be regulated under the current Biosafety Act and the regulations. So those would be genome edited organisms that would lead to a GMO. Now, for those that do not have any genetic material, any foreign genetic material, we have provided a different pathway of regulation. So, as I just want to confirm, genome edited organisms will be regulated using two pathways. And those two pathways have been elaborated very clearly in our guidelines that we published in March.
Okay, thank you for that clarification. Indeed, because many scientists would do their research, and at the end of the day, they would not have any foreign DNA within the product that they come up with. So they would have to follow a different pathway as compared to others that would have the foreign DNA remain within the product. It's good to clarify that to the various participants that we have. So I'll go back to. Yes.
I wanted to add something maybe just to provide more clarity. We have provided those two pathways. So the guidelines also provide for the scientific community to know whether they are regulated in either of those two pathways. We have provided a process called the early consultation framework. So all scientists or any person who is intended to do any activities involving gene editing of plants, animals, or microorganisms, they are supposed to approach NBA. We have provided a form which we are calling the early consultation form. So do naturally fill that form, and that form will be reviewed internally, and a decision will be made within 30 days. So within 30 days, NBA should be able to provide a very clear framework or a very clear direction on whether the gene-edited organism or product that they are working on follows under the regulation of NBA under the Biosafety Act.
Okay, thank you for that clarification. I think it makes it much easier for our participants to also understand that there is a transparent process that the NBA has, and indeed open door policy as well, to ensure that you have a dialogue before the research commences, where you engage one-on-one to identify which pathway the various research they're proposing to do would follow through. I want to go back to you, Dr. Tripathi. You published a paper recently in partnership with Professor Runo here, which was titled Genome Editing for Sustainable Agriculture in Africa, which I very much loved reading, and it was quite insightful. What are some of the highlights you would want to share with us from that in a more continental approach and of what genome editing stands to offer?
Thanks, Doris, for this interesting question. So basically, the whole idea of publishing that article was like, to say that, genome editing is a powerful technology which can be used for the sustainable agriculture. And basically, that, the population is growing. So currently, the major global challenge is how to feed the increasing population. And when it comes to Africa, actually, the population in Africa is predicted to be doubled by 2050. So that means, like, even agriculture production needs to be doubled, but we don't have the increase in resources. So it's still on the same similar resources, which we already have. And now, so that means, like, I'm not saying that, gene editing is a silver bullet which can solve all the problem. No, we are not saying that. What we are saying is that genome editing is one of the powerful tools in the toolbox. And when it comes to the urgency or to the critical need, because right now the critical need is to minimize the yield gap, what is there in all the staple food crops, whether it's maize or is sorghum or is banana or is cassava. So, then you have to decide, which tool can be used to solve one or two problems, so that's how we feel like, in the toolbox, one another technology has been added, which we have as a scientist, we have access to that. And basically this technology is easy to adapt because it's simple, is quite precise, targeted, and it's like, any lab which can do the molecular biology and the tissue culture, those type of labs can easily apply this technology. What you need is like a reference genome of a crop, some bioinformatics tool, and some of the laboratory facilities. So that's some of the things we were talking about in that paper. And, we also talked about what are the ongoing projects, so it's not that everything is going outside of Africa. No, within Africa, a lot of projects are going, and some of the projects, products are in the pipeline. For example, like Steve talked about the sorghum, the striga-resistant sorghum, that's in Kenya and is ready for the field trial. Then there is a maize, which is maize has been developed for resistance to the lethal necrosis disease. And then, that is like, is also, that project is going on in Kenya. And then there is banana project, as I mentioned that, we are working on several diseases with banana project. And there is a wheat project, which is more in northern of Africa, which is Egypt. And then there is a cassava project going on in several countries, like in Kenya, in South Africa, as well as in Nigeria. And then as Professor Muchiri was talking about, like, means, okay, if the technology is there, and then the enabling environment is not there, technology doesn't make any impact. So, so it's very necessary that, technology should make the impact. And for that one, we always need the support from the regulatory. So, and Africa is actually moving very fast as the globally countries are moving. So in two countries, the regulation, or I should say the regulatory guideline for genome-edited products are already in place. That is, Nigeria was the first country, and then recently Kenya approved those guidelines. And that also shows that, that people have confidence in these technologies. And there are several other countries in Africa which are currently under discussion to also developing the guidelines for gene editing. So these are some of the highlights of that paper. And we also talked about there are some other crops which are the staple food, particularly in Africa. And then if the scientists in Africa don't work on those crops, who is going to work on those ones? So, we have to work, because as I said, like, most of the places, cereal crops are the crops which are feeding more people. But when you come to the East Africa, actually, there are vegetatively propagated crops like bananas and cassava, which are feeding more people in comparison to the other cereal crops. So, so we have to actually close the yield gap in these type of crops as well.
Yeah. Thank you. Thank you for those highlights. And indeed, I noted there were 3 tuber crops and 3 cereal crops. So very well balanced with that paper. And thank you for sharing the highlights, even around the regulatory system, as you say it. I'll go back to you, Muchiri. How many other researchers is the NBA regulating currently on genome editing, aside from Leena's research and Professor Runo's research? Is there anything else?
Yes. As NBA, we have approved since 2015. The first application for genome edited we received was allowed 2014, but the decisions were made in 2015. So far, we have approved. The board has considered and approved 10 genome edited products or projects, but all of them are basically at the laboratory at the greenhouse level. Most of these projects are basically at EDD and SIP. but a number of other projects are also at Kenyatta University, as Professor Runo has just indicated. So we have approved a number of crops like potatoes, yams, bananas, grass pea, and a number of other crops which are being edited for a number of production constraints like agronomic challenges. And also we also have about 1 or 2 projects targeting animal-related production constraints, and one for vaccine development. So far we have approved about 10, but all of them are still at the basic laboratory and greenhouse phase.
Okay, thank you for that. And indeed, I think the list will be growing even as we advance, because this is a technology that we have more to offer. And as we heard from the benefits, it's precise, it's specific, and it's it's also quick to implement. I want to come back to you, Professor Runo, in terms of the research that you've been doing at Kenyatta University around striga and genome editing. How has your experience been when it comes to the regulatory process? When was your approval granted, and how has that been with the regulator?
Yeah, before I answer that question, Doris, I think there's a question on the chat that maybe Leena will address in a few, and it comes from Professor Johnny Gressel. Good to see you, John.
Yeah.
One of my greatest mentors and somebody I look up to. Also a parasitic plants research. So the question is,
so maybe Steve, you can answer Doris' question first, and then I will take that question. I have seen that question in the chat. Yeah.
Okay.
Yeah.
All right. So yeah, so the question that Doris asked is what our experience has been dealing with regulatory authorities, and I think for us, and I think for Kenya in general, and I hope most of Africa will follow suit. We have a very competent National Biosafety Authority. So the team we have at NBA is technically sound and very approachable, with very clear systems and frameworks that one follows. So in our applications, not just for genome edited applications this year, but for further applications of biotechnology and genetic engineering related, we have had a very straightforward, very good and pleasant experience dealing with the National Biosafety Authority. So thank you. And I think I'd be doing injustice not to commend the NBA for the way it has made it easy for us to do science. So in short, I think we have had a very pleasant experience. They're very professional, they're very knowledgeable, and they're also very willing to help and listen to us.
Thank you for that. And I'm seeing Leena also nodding, so I believe it's a similar experience with you as well. I think that is very good feedback for future researchers who would want to engage with the authority. Ladies and gentlemen, as you see, we are way past time. I'm also looking at the various chat boxes to see the various questions that you've been sharing with us. We ask that you continue engaging with us with your questions. And as Professor Runo highlighted, there was one question here for you, Leena. Jonathan is asking, it was not clear to him whether you are transferring the Xanthomonas gene from the wild using gene editing as an insertion tool, or you'll be using it to modify the endogenous genes. If it's the latter, what use was the wild species data and how was it used? Yeah.
Thanks, Jonathan, for this question. And sorry, I was not providing much technical detail before. So basically, as I was mentioning before, only the Musa balbisiana, which is a wild progenitor, has the resistance to the bacterial disease, and all other cultivated varieties of banana have the are all the susceptible. So I was very curious to know what makes the balbisiana resistance. So we did a comparatively transcriptome analysis, comparing the early infection period after infection with the Xanthomonas to the Musa balbisiana with a most susceptible variety. And based on that, we find out several target genes which were differentially regulated upon pathogen infection. And I was mainly interested at the early infection, because we have to control at the early infection site. So we found some of the genes overexpression, and then some actually was downregulation. We were more interested in the overexpression, and majority of them were actually either the susceptibility genes or the genes which are involved in the transportation of sugars or nitrogen. So, you know, we are actually knocking out those type of genes. So those are the endogenous banana genes, because we align the sequence from the banana variety we have tested with the varieties we are now doing gene editing, and we design the guide RNA from the conserved region. And that's how actually, that's the approach we are following. The second approach we are also using is that based on the literature, if we are trying to see the susceptibility genes, like one of the paper we have published is the DMR6, downy mildew resistance 6 gene, and so we have actually identified the homologue of that gene in banana, and then we try to knock out that gene, and it has given us very good results. So we have been testing more than 10 different susceptibility genes in our lab, and those are all knockouts, so that we don't have any foreign gene to bring from the outside, from the bacteria or from any other plant species. I hope I have answered your question.
Yeah, I believe so. Thank you for that, Leena. I don't see any other questions, so I'll continue with the questions that I have here. And I want us to talk a bit about the impact, especially when it comes to the farmers and the consumers, because you've highlighted as you introduced the research work that you're doing. They're the ones who stand to gain the maximum benefit from this research. So one of the questions that came about is that since work that you're doing is around the public sector work, will the crops you're working on be offered free or at a low cost to the various farmers? I can direct it to Runo or to you, Leena.
Yeah, I can answer because I'm also in the public sector. So the research we are doing, if we are developing a disease-resistant varieties, definitely those varieties will be available to the farmers with no additional cost. So if they are paying for the sucker, like let's say 100 shilling, they will still pay the 100 shilling to get that sucker. Or if they are buying the tissue culture plantlet at whatever rate, they will still, so they will not pay anything extra for these varieties, because the varieties are developed through the public sector. Yeah, that's in the short answer. Yeah.
Thank you for that clarification. And for you, Runo, will the sorghum be free, or will it be at the same cost for farmers?
Well, there is so we are not in this for profit. So let me say that that's first. So no, we are not going in for this to increase profit margins. No. But at this point, we are not very clear on how much it is that has been invested in terms of research and development. So we have not costed that. So it's really hard at this stage to say how much it is and if that's going to be more or less than what farmers are already purchasing.
Okay, thank you for that clarification.
Doris, if I can also add, so like if so there will be these varieties will be freely available to the farmers. So that means, like, if they want to get a sucker from their farmer's field, or from their friend, or they want to share their varieties with, so they are allowed to do all those things. So we are not going to regulate those things. Yeah.
Wonderful. Wonderful. And I've seen one farmer joining us, so I'm sure that is very good news for him to hear that from you. I want to come back to you, Muchiri. Last week, we had some exciting news around genome editing regulation from the UK, where they're relaxing their law on the genome editing research. I want to ask you on a more personal level, maybe a more national level, is this good news for Kenya? And if it is, why?
Okay, thank you. Thank you, Doris. As you may know, the issue of genome editing is not very old. It's not old. Different countries are at different stages of developing their frameworks of how they intend to regulate gene editing. Like you said in your introduction, in Africa, I think only Kenya and Nigeria have been able to publish very clear guidelines on how genome editing technologies are regulated in those countries. Other countries, I know they are still developing. Maybe very soon they're also going to have their own country-specific guidelines. Now, development of or approval or relaxation of rules, I think in developed countries is a welcome move because, as you know, even research is done in collaboration. I know Professor Runo and Leena, when they are doing research, they don't do it alone here in Kenya. They collaborate with other institutions in other countries. So if rules are more or less harmonized to a large extent, that would be able to facilitate research and maybe possible commercialization in the future.
Thank you for that encouragement as well. I want to ask this question. I think it applies to all of you in terms of policy. What can be done to ensure that these technologies actually reach the beneficiaries? Prof, you're nodding. Would you want to take that one?
So I think from the perspective that our agriculture is very, in general, or in the context of African agriculture, I think there's a lot of players, there are a lot of stakeholders. So it's not A leads to B, leads to C, leads to D. There are policies that need to be very well laid out. There has to be investment in terms of the government, in terms of extension. There has to be involvement, and there has to be value for the product that you're developing. So, for example, if you're developing a product that is gene edited, it has to have a specific value to a farmer. Is it something that they will be happy to consume? Is it in science or in technical terms, to say, in the right kind of genetic background? So that will enhance technology uptake. Then there are other issues that are also very unique in the context of Africa. So the land tenure system, is it ideal and is it appropriate? And issues of gender, what the male gender prefers, because they are the custodians of the land in these cases, but then also the female gender. So all these need to come into play, and all this has to be very well thought through for you to develop and commercialize a product. And then most importantly, what we need to think about is our seed systems, because for for the most part, we don't have, I think in Kenya, I can speak about Kenya, the seed systems are not very optimal. So I think we'll need to think through this and all these small things that will eventually lead to successful uptake, commercialization, and adoption. And within this, there has to be a very good policy, and there has to be very clear understanding and very transparent communication all through the process. So it's a lot of things that need to come together for appropriate and successful delivery.
Yes, Leena, you want to add?
Yeah. Yes, I want to add. So first of all, if we really want to have the maximum benefit of this technology, then we need an enabling environment in Africa. I know some of the countries have already developed policies, but many of the countries doesn't have policies yet. So even though the countries are developing policies, you will see that some of the policies are not the same. They're a bit different as well. So what we require actually, if you want the maximum benefit for the Africa, if you're talking about, then is like a harmonization of the policies, because the countries, the borders are porous, and if you're let's say if tomorrow if we have bananas in Kenya, and then whether you will just restrict those ones to Kenya, you won't like that the farmers who are suffering from the same disease in Uganda should also get benefits of those bananas in Uganda or in Tanzania or in Rwanda. So I think that's what the harmonization of the policies are must. The second thing is that the technologies, the IP issues needs to be rectified as well, because like the CRISPR-Cas, whether that's freely available or what. So I think that's the another thing. And then the another aspect is public awareness, the information to the public, because the public is still not everyone is very clear about the differences between the genetic modification and the gene editing, and so they are confused whether the gene edited are still GMO. But there is a big difference that the gene edited product can be GMOs if the foreign gene is integrated in them, and if they have no foreign gene integration, then they are non-GMO. So I think this type of differences needs to be public needs to understand that, so that there is no fear for any adaptation, because whenever there is a new technology, there is always a fear associated with that. So a lot needs to be done with the advocacy, the public awareness, with the science communication. And I'll say so science needs to be in the center. So the policies should be science-based, the communication should be science-based for the public in order to get the maximum benefit of this technology in Africa. Yeah, I'll stop here.
Wonderful. I wouldn't even want to add anything more to that. I think both you and Prof have complemented it very well in terms of what we would need to have for an enabling policy environment around this technology. I see we are also coming close to an end with the time. I haven't seen any more questions coming in. So I'll ask one more question and then we can go around in giving the parting shot from each one of you. So the last question that I have here is a more personal question in terms of what it is you envision as the future of gene editing technology, especially to the consumer and as well as to the economy. What should we look out for? So I can start with you, Leena.
So I will say that, particularly for the diseases where there are huge economic impact, like for example, the bacterial disease has made between $2 to $8 billion losses in East Africa, and I would like to see the disease-resistant bananas closer to the farmers so that those losses can be minimized. And then we would definitely love to see the gene edited crops are treated similar to the varieties which are developed by any other technologies, like whether it is breeding or it is a chemical mutagenesis or others. So those varieties are treated. If there is no foreign gene integration in them, then they should be treated similar to that. That's what I would like to see for Africa. Thanks, Doris.
Wonderful, wonderful. Thank you for that. So we treat gene-edited technology similar to conventional. Thank you. Prof, what do you see as the future for gene editing technology, especially to the consumers or as well to the economy of the country?
Yeah, so I think that's an important question, a bit complex. But here's the thing. So there's been a lot of investment prior, or we'll need a lot of investment prior in terms of knowing what genome editing can do for improvement. So I think the crops or the plants that have had a lot of research done on them, and these are, I'm calling low-hanging fruits, and traits that are easy to improve with genome editing. As Leena said in the beginning, it's not true that genome editing can improve all plants or all crops for all traits. So there's certain limitations with genome editing. And I want to point out, for example, what to look out for would be, for example, virus resistance, because these are targets that you can be able to develop quite efficiently. There are nutritional improvements. So for example, you can be able to, you should watch out for reduced cyanide in cassava, for example. Should look out for enhanced digestibility in sorghum, because the way that genome editing works is that, at least for now, is that it works on removing the function of genes, or what in technical science terms we call gene knockouts, or inactivation of genes, or removal of adverse gene sequences. So the ones that have had quite a bit of investment prior, and the ones that are easy to target with genome editing. So low cyanide cassava, disease resistance in terms of viruses, and improvement of nutrition in sorghum, removing anti-nutrients and increasing digestibility. And of course, striga-resistance.
Thank you for that, Prof. I think you're giving Mr. Muchiri a reason to know that he has more work coming in the future on his plate. Muchiri, what do you see as the future of this technology to the consumer and to our economy as well? Is this going to be a money maker for Kenya?
Thank you very much, Doris. From where we sit, the future of the technology seems quite positive, particularly on the technologies that ensures that there is no integration of foreign genetic material. As you know, one of the biggest concerns in terms of genetically modified organisms is basically the integration of foreign materials. There has been a lot of concerns on whether that integration would have some adverse effect in the future. So when you're looking at these technologies, the genome edited, and assurance by the scientific community that there is no integration of foreign genetic material, it gives a lot of confidence, basically to the consumers and also the policymakers. From where we sit as regulators, I think we will continue providing facilitative regulatory frameworks in terms of this technology, because as the technology advances, we also need to assure the public the products that are being channeled out from that process are safe for human consumption. That is a space that will continue guarding even as science goes forward. Thank you very much.
Thank you very much. Ladies and gentlemen, we've come to an end of this webinar session. Indeed, it has been super enlightening and a very engaging conversation. We've heard from the various researchers the kind of work that they are doing, and to the specific of why they are doing it and what it stands to benefit to the various consumers and and to the economy as well. We've also heard from our regulator as well in terms of how they have decided to approach this technology and offer an enabling environment for regulating. So thank you very much to all of you, our wonderful participants, for the joining us and creating time to be with us. We urge you to continue this discussion on our social media pages. As we said, we would find this discussion continuing on the Twitter, Facebook, as well as Instagram, and the handle is @ScienceAlly. I also want to thank especially our remarkable scientists for providing us with the great updates on the status of gene-edited crops in Kenya. I'm particularly excited and look forward to the commercialization of some of these products myself. And as they were said, they'll be free. I hope there will be also not free, but same cost as the counterpart conventional crops as well. So with this, I'd like to thank you once again for tuning in, and see you next time. Do enjoy the rest of your day. Goodbye.
Bye.
Thanks. Bye. Thanks, Doris, for moderating it.
Thank you.
Bye, thank you. Happy Madaraka Day, everybody.
Yeah.
In advance.
Okay.